Good afternoon, everyone, and thank you for joining us for today's webinar. We do apologize for the late entry, but we are going to get started here in just a couple of minutes. Before we do get started, I would like to go over a few housekeeping items with those of you that have already joined us in the room. For those of you that we do have in the room, the first thing I would like to make mention of is our chat pod, which is located on the left-hand side of your screen marked chat. This chat pod will be the best way to communicate with us should you have any questions regarding today's material. All questions pertaining to the material will be retained until the Q&A session that we will have at the very end of today's webinar. So please feel free at any point of today's discussion to go ahead and type in your questions there in the chat pod. We will pull those out and house them for you, and then we will get them answered of our subject matter experts directly after today's presentation. This chat pod is also the best way to communicate with us should you experience any technical issues. If you do have any technical issues, please make sure to let us know there in the chat pod, and either myself or one of our customer service representatives will make sure to get you taken care of. All of your audio for today's webinar is going to be streaming through your computer speakers, so if at any point of today's talk you do happen to lose your audio or the live video feed of today's presentation, the best thing that we can suggest for you to do to troubleshoot is to simply refresh your web browser and then hit the play button again in your live feed window. This should refresh your connection and allow you to rejoin the live audio and video of today's talk. Also, if you do experience any other technical issues, or if for some reason that does not work, we do have our toll-free number as well as our email address that are located directly above the live feed window, should you want to use that mode of communication with us to get your troubleshooting worked on. Also below the live feed, we do have the closed captions pod as well. So make sure that if you would like to follow along in our closed captions, you are doing so directly below the live feed. We are going to get started here in just a couple of minutes. I'm going to switch some things up. I have seen that a lot of you have already typed in your location there in the chat pod. For those of you that have not, if you could take a few moments before we do get started to type the location that you are watching today's event from, that'll give us some fantastic feedback about our audience as well as the kind of reach that we were able to obtain with today's webinar. Again, we are going to get started here momentarily. I'm going to switch some things up, and then I will come back on to introduce our speakers for today. Thank you for joining us. Good afternoon, everyone, and thank you for joining us for today's webinar. My name is Josh Vickers, and I'm with RTI International's Forensic Technology Center of Excellence. Today's webinar is entitled "The Time to Analyze Is Now: DNA Evidence in Groping Sexual Assault Cases." We have two fantastic presenters today. Our first subject matter expert and presenter is Dr. Julie Valentine. She is the assistant professor at Brigham Young University, BYU College of Nursing, and a certified adult adolescent sexual assault nurse with Wasatch Forensic Nurses. Her clinical specialty and research focus areas are sexual violence, intimate partner violence, violence against women, and criminal justice system response to sexual violence. Dr. Valentine focuses on multidisciplinary collaborative research studies, uniting disciplines in sexual assault case reform to benefit victims and case processing. Our second speaker and subject matter expert is Ms. Heather Mills. Heather Mills has been a forensic scientist for the Utah Bureau of Forensic Services for over 9 years now. She is proficient in serology and DNA analysis, lab work, as well as interpretation, and currently is the manager of the homicide team, which is made up of analysts that perform casework in serology, DNA lab work, and DNA interpretation. She is the serology technical leader and is also an alternate member of the laboratory crime scene response team. Just our quick disclaimer before we get started: the opinions, findings, and conclusions or recommendations expressed in this presentation are those of the presenter and do not necessarily reflect those of the Department of Justice. Any products and manufacturers discussed are presented for informational purposes only and do not constitute product approval or endorsement by the U.S. Department of Justice. And now, without further ado, I would like to hand things over to our first speaker of the day, Dr. Julie Valentine. Dr. Valentine?
Welcome. We are happy that many of you are able to join us on the webinar. We are going to just switch over to another screen as we begin. The title of this webinar is "The Time to Analyze is Now." This webinar builds upon a prior webinar that was done in February 2019. And the title of that webinar was "The Time to Collect is Now," and also was focused on groping sexual assault cases, the use of touch DNA in sexual assault cases. We had a huge response following that webinar. The focus for that one was really making a case for both our criminal justice system partners and for our forensic evidence collectors, sexual assault nurse examiners, medical forensic examiners, on the importance of collecting DNA evidence in groping sexual assault cases and also collecting touch DNA in sexual assault cases. Following that webinar, we had a significant response, many questions, and many of those questions focused on forensic science aspects about the analysis of the touch DNA. So this is a second webinar that's meant to build upon that earlier webinar. For your reference, we have uploaded a PDF of that first webinar. We've also uploaded a PDF of literature review or references for you to use. And we've also uploaded a PDF of our State of Utah Touch DNA form that we use in groping sexual assault cases or other sexual assault cases, if you find that helpful for your jurisdiction. So without further ado, we'll get started on somewhat of a review, and then Heather Mills will begin our information on the analysis portion. Next slide. And we will move forward to the next slide. The next slide is slowly coming up. We have been having some internet connection issues, so bear with us momentarily. In Utah, we are in the midst of a snowstorm. And unfortunately, it's difficult to control weather and sometimes how that might affect our internet. I'm just going to go ahead and start talking about some information, and then that next slide will be coming soon. So I'm going to be reviewing collection techniques and really be pulling some of the information from the document released in August 2017 from the National Institute of Justice, the National Best Practices for Sexual Assault Cases, a multidisciplinary approach. We're going to talk about the importance of clean techniques, both at the time of evidence collection and also in the lab. And this is especially important when we're talking about touch DNA. Sometimes the term touch DNA and trace DNA are used interchangeably, even though they are not exactly meaning the same thing. But with touch DNA, we generally are talking about small amounts of DNA. And it's very important that we do not contaminate that sample, since we are really focused on those smaller amounts of DNA. And then Heather is going to be talking about introduction to the lab processes and procedures. And she has geared this presentation not only to be helpful to forensic scientists and talking about validations and trace DNA and mixture interpretation basics, but also for our multidisciplinary partners so that they will have a better understanding of what occurs within the laboratory on these cases with trace DNA or touch DNA. In the earlier presentation that we did last year, we provided a case study that we will be referencing again towards the end of this presentation. If you'd like more information, specific data on that case study, please pull up that earlier webinar. The earlier webinar also has research finding from cases that we have that have developed helpful DNA from groping sexual assaults. And at the end, we will leave time for questions as well. So feel free to send those questions in. Next slide. So I would like to discuss the application of touch DNA in sexual assault cases. When we think of touch DNA, one of the first things that comes to mind is sexual assault or groping cases by strangers. Much more applicable in those cases. If somebody is sexually assaulted by an acquaintance, a boyfriend, it might be very likely that we are expecting to see some of their DNA on the person, but that also is dependent upon where on the victim. So groping sexual assault cases and the case study that we will be discussing is focused on a groping sexual assault or sexual assault by a stranger. Then we have groping sexual assaults by known individuals dependent upon the body location. If it is a known individual, but we're finding touch DNA on a genital area, and this can be especially very helpful in pediatric cases, then using that touch DNA can be very beneficial to the case. And then not just groping sexual assaults by strangers, but sexual assaults by strangers in general. Sexual assaults by multiple perpetrators. I had a case where a woman was raped by two men. One of the men held down her upper body, her arms and shoulders, while she was vaginally penetrated by the other assailant. So I collected evidence from both her upper body as suspect one and collected evidence where the contact was from the perpetrator that vaginally assaulted her. And then also they can be helpful in sexual assaults or contact by perpetrators with protective orders. If we find DNA no contact order, we should not find that DNA on the victim. Then there's also, can be helpful with touch DNA. We are doing more and more exams on assailants or suspects, and this can be very helpful if we find victim DNA on assailant or suspect, and this also could include touch DNA. Next slide. So a quick review on touch DNA and what impacts the amount of touch DNA left on an object or skin on somebody's body. Donor sheddar status is high on the list here. Now, most research studies, and again, you have that reference list if you would like to refer to some of these, most research studies have found that we have some individuals are what we would call high shedders and some are low shedders. And this generally is fairly consistent. There have been a couple research studies that have stated we may not know if this is fairly consistent because other factors may be at play, which I'll go on to later regarding personal habits. But overall, donor sheddar status is something that we see as a big impact on DNA left. Gender. Males generally are higher shedders than females. Age of males, Younger males between the ages of 20 and 40 are often seen as the highest shedder group. And then personal habits make a difference. If somebody has recently washed their hands, we are more likely to see less touch DNA. If someone has a personal habit of rubbing their face or parts of their body and then touches somebody, then they are likely also picking up epithelial cells from those other parts of their body and will have more touch DNA. Sweaty or dry hands. So if someone's hands, an assailant's hands, are sweaty, there is more touch DNA passed to an object or a person through that sweat. But we also see it with dry hands, that flaking skin. And then the surface of an object makes a difference. If the object is porous or non-porous, textured or smooth, those objects that are textured are more likely to collect DNA. And we can look at that when we look at the research studies that have developed touch DNA from clothing. Most clothing is textured, and in our case study, the clothing on the part of the clothing that was textured was where we developed a STR DNA profile that was from touch DNA on that textured part of the fabric. And then the type of contact makes a difference. If it's a short amount of contact, if it's a brief touch or a light touch, compared to if a person's arm is grabbed, whether clothing or on the skin, and held tightly, or if there's friction during that contact, which frequently can happen if a victim is working to get away from the assault. They might be pulling against the hand, and that hand might be rubbing back and forth. We then are more likely also to find touch DNA. So next slide. So I referred at the beginning that we would be talking about the national best practices for sexual assault kits and multidisciplinary approach. I hope you're all very familiar with this document, a very important document. But our approach in collecting touch DNA is focused on the concepts presented in this document. So we try to concentrate the collection by using no more than 2 swabs per area so that we do not dilute the biological sample. The focus is to concentrate as much DNA as possible within those 2 swabs. If swabs are collected from a skin, they should be lightly moistened, and 2 swabs generally held together and collected together. If the swabs are overly wet, you may have to do wet and then dry. If your jurisdiction does like wet and then dry swabs, please note that which ones were wet or which are dry. But generally, the movement right now is to have 2 lightly moistened swabs on that skin area. We also recommend that rather than examiners collecting from the clothes, that the clothing be packaged and sent to the crime lab, clearly designating where the touch occurred, but the examiners would collect from the body area. Next slide. This next slide also shows directly from the National Best Practices for Sexual Assault Kit. It clearly states skin for touch DNA. This document is the first document that has clearly stated on national best practices that we should be collecting touch DNA from skin. And again, it states use 2 lightly moistened swabs across the affected area. And next slide. Now, to avoid cross-contamination, as I talked at the beginning, when we're looking at smaller amount of DNA, incredibly important that we keep our sample as pure as possible. So from the national best practices, this includes using single-use tools and lubricants. We generally do not use any kind of lubricant except sterile water for skin. We limit the number of people in the room. Of course, our approach always is to be patient or victim-centered, but especially at the time that we are collecting the evidence, we need to have the support person not breathing over or close to those collected swabs. At the time those swabs are moistened and then evidence is collected from our patient or victim is a time where we really can run the risk of additional DNA going to those moistened swabs. So gloves and mask worn on the examiner during evidence collection, and we should avoid talking over any of these swabs or talking avoided by the examiner during the short time of the evidence collection and packaging. We especially need to be very cautious when the support person is a male, because we are looking, they will be looking oftentimes, if it's a female victim and a male perpetrator, they will be looking for the male DNA. So just take that case by case and be very cautious to get a pure sample as possible and avoid cross-contamination. And next slide. And with that, quick review, and again, I will refer you to the earlier webinar, and we also will answer questions at the end. I'm going to turn the time over to Heather to give the perspective from the forensic scientists and lab perspective.
Thank you, Julie. So next slide. So I want to give a little bit of a disclaimer before we get started too in depth. I just want to note that all forensic laboratories are set up differently, and they have a variety of different instrumentations, methods, procedures, processes to run their samples. And so the rest of the presentation will give you a general idea of how our lab is set up, how we do it, but it is by no means the only way or the best way. And each lab has different and unique requirements that they use to meet their own community, the needs in their own communities. And so we encourage you to work closely with your local laboratories and learn their processes, ask questions. Those relationships are really important for the overall success of the entire process.
My license has page press filter on it, but my legal version.
So in addition to that, new technologies are consistently being introduced and implemented for forensic use. And each lab is responsible for deciding what works best for them. So we really encourage you to develop those relationships. Next. So I want to start off by discussing some of the clean techniques that are necessary when working with DNA in a forensic environment. And then we're going to talk a little bit about separation of lab space and why that's important. And then we can continue with what Julie talked about with proper personal protective equipment, or PPE. So when processing samples for DNA, regardless of whether we're working with trace touch DNA samples or if there's a biological fluid present, clean techniques are necessary in order to help you prevent contamination. Any lab spaces that are used for body fluid testing or DNA analysis should be considered clean labs. That means that anybody who enters the lab should be putting on their personal protective equipment before they enter that lab space. And that personal protective equipment is your first line of defense for preventing contamination. So minimum PPE requirements would be a clean lab coat, a face mask, and gloves. And then you should change out your lab coats and your face masks frequently, and at a minimum in between each item of evidence. or anytime you feel like your gloves or your mask might be compromised. Next slide. So here's kind of some examples of our setup. There on the bottom left is where you'd enter the lab, and you have space to put on gloves and masks, wash your hands, do that kind of thing before you enter into the lab. The picture on the far right is an exit of our lab where you can wash your hands, dispose of your PPE before you enter into common laboratory spaces. And then additional techniques that should be employed when working in the lab. You should make sure to be cleaning your lab benches and your workspaces with, we use 10% bleach, but bleach before and after any sort of analysis. Put down a barrier to maintain a clean workspace, like butcher paper or something like that. You should change that paper in between every item of evidence. And then all of the tools that you use to cut or prepare samples for DNA need to be UV sterilized frequently, cleaned with bleach, and with ethanol in between every item. Next slide. This is kind of an example of a setup of a lab. We have a serology lab space where you can do biological fluid testing, and then entrances into the areas where we do DNA analysis. Your extraction area, we're going to talk a little bit more in detail about each of the steps, but the extraction area and the pre-amp area are separate from any post-amp DNA. And so that red circle, that area has a one-way flow, meaning that you can only enter and exit from one way. You can't go back into those other spaces. It's an exit only so that you don't get contamination from your DNA samples into other areas of the laboratory. And so the lab design can help prevent and minimize contamination. So in addition to wearing proper PPE, the access to this laboratory space should be limited. You can maintain an employee database of the DNA profiles of all the employees who work in this area and work in this space. And that database can and should include DNA profiles of any maintenance personnel or anybody who enters that lab space, which can help you when you get to your DNA interpretation of your samples. Next slide. So these are just a few more pictures of how that lab space actually visibly looks. That bottom left is a picture of where we're going to do extraction. So we'll talk about that in a few slides. Middle is where we set up our DNA samples before they get amplified. The bottom middle is a picture of what we use to amplify or make copies of the DNA. The top right is a picture of a pass-through. So we actually pass our plates through without walking them through the lab space. And then the bottom right picture is a picture of the post-amp room where all of the DNA stays after it's been copied. Next slide. So I'm going to talk a little bit about case flow, how samples come into the lab and what the procedure and process for that is. When a sample is submitted to the lab for DNA testing, it's generally submitted to an evidence section for testing. Here, everything that comes into the lab comes in for a biology assignment, and it will go through the serology section first. Serology is where we test for biological fluids. We can swab items for DNA, cut samples for DNA, and then we'll issue a report if we do test for biological fluids. Those samples that are deemed necessary are sent on to DNA, where we try and individualize those stains or material that might be present. And then a report is issued describing those DNA results. Next slide. This is a little representation, a little bit of the DNA workflow. Once something comes through serology and is sent on for DNA, This is a descriptor showing two different pathways. One might be a robotic pathway or one might be a manual pathway. The robotic pathway, the lab work is done by robots, which can help reduce contamination, minimize sample handling errors, things like that. The robots are set up and run by DNA analysts. In the manual method, DNA lab work is done by hand, manually, by analysts, meaning the analysts are the ones opening the tubes, transferring liquids instead of robots. Next slide. So here's a picture of the processes that we're going to talk about a little bit more in depth. This is a visual representation of what a manual process could look like. Once a sample is sent into the DNA section for DNA analysis, the first step that it goes through is called extraction. The extraction is going to remove the DNA from the stain or the substrate that it's on. When dealing with trace DNA samples, touch DNA, it's often off of swabs. So we're trying to get the DNA off of that swab and into a solution. This step also cleans that DNA, cleans it up, all the proteins and any cellular debris that might be present. It purifies it so that we can work with it better. Once we can get that sample into solution, it moves on to a quantitation step. And this is the instrument that can do quantitation. That's going to determine how much DNA is in our sample. Once we know how much DNA we have, we can move along into the process and make copies of the locations that we want to test for. And then we can put it onto an instrument that's going to separate those locations on the DNA strand by size. And then we can generate what's called an electrophoregram that is going to be used for DNA interpretation. So I'm going to talk about each of these steps in the next few slides in a little bit more detail. When you do lab work manually, all of this plate setup, extraction is all done by hand using manual pipettes and pipetting into plates, wells, things like that. It's all done manually by an analyst. Next slide. Another lab process that could be robotic, it's all of these same steps, but done with robots that can pipette for you to handle more samples. And so all of those steps are the same that we're going to talk about, but that close-up picture in the bottom of your screen shows you that the robots are actually doing that pipetting instead of an analyst. Next slide. So the first step that I want to talk about is extraction. So like I said, the purpose of the extraction is to remove any potential DNA off of the substrate or the stain that you might be working with. And it's also going to purify the DNA that is present. It's really the first step in the DNA process. There's many different kinds of extractions that can be used in forensic DNA. And lots of labs use different kinds. There's organics, there's chelax, there's different robotic extractions. There's a wide variety. And so, like I said, we encourage you to work with your labs to find out what they do and how they work with their extraction process. But once the DNA gets into solution and it gets purified, cleaned it of any other debris, dirt, cellular debris, proteins that can degrade the DNA, those are the kind of things that we're trying to get out of our sample so that it doesn't degrade our DNA downstream. And all samples, if they've gone through serology and they have blood or saliva or working with trace DNA, they go through this process. When working with sexual assault samples that might have seminal fluid, there are some additional steps that are employed during the extraction process. The purpose of that is to separate any possible sperm cells that might be there from the rest of the skin or epithelial cells that can be found in seminal fluid. This additional step is called a differential separation, and it generally turns one DNA sample into two. So you'll have, for example, vaginal swabs, sperm fraction, and vaginal swabs, non-sperm or epithelial fraction. Next slide. So once we've extracted the DNA, we've got it all cleaned up, it's ready to go. We have it in solution. The next thing we're going to want to do is find out how much DNA is in our sample. Quantitation can also help us check for any remaining inhibition or issues that we have with our sample. And it can also give us a male-female ratio. So once we know how much DNA is in our sample, that's going to allow us to make a lot of decisions in order to optimize the rest of the lab process. A lot of the decisions made with forensic DNA are made after this quantitation step. In some labs, if there's no DNA present or there's no male DNA, depending on the type of case, it may stop at quant and no further analysis will be done. But that is going to depend on the lab and the validations that they've done, which we'll talk about a little bit later as well. So the case scenario or the victim history can help us determine how to process samples and figure out what kind of testing to do. So labs that utilize YSTR testing, some of those decisions might be made at this step as well. So what route are you going to take it through the lab? What kind of testing are you going to do? And how is that going to help you with your sample or get you the information that you need? And that's where that male-female ratio can come into play. It can be useful in a lot of cases, and especially sex assault cases, to help determine if YSTR testing would be beneficial or regular SER testing is the route to go. We'll talk about that a little bit more. For trace DNA samples specifically, it is common to have low amounts of DNA. So knowing this at quant can help you decide how much of your sample you're going to need to use. Are you going to have enough DNA? Should you be trying to target a higher amount at the next step in order to work with a low-level sample or something that might have a mixture of a lot of people? So this quant reaction can help clue the analyst into any possible degradation issues or things like that that you might be dealing with when you're dealing with low-level samples that are commonly seen with touch or trace DNA. Next slide. So once we know how much DNA is in our sample, now we want to do what's called normalization. During this step, we're going to use the amount of DNA and we're going to either dilute or concentrate the samples to get to the same target or amount of DNA for DNA amplification, which is the next step. This is done in order to optimize that amount of DNA that we know that we need for the best, to get the best amplification, to get the best profile in order to make it interpretation easier. So these steps and amounts and all of this process is based on studies that the labs do with their instrumentation and their reagents to determine what's going to work in their system. So based on these studies, you can determine what your target amount of DNA should be and what you should normalize your samples to. When working with low-level DNA samples, sometimes you can target a higher amount, or you might have to concentrate it all and take all of the DNA from that sample to try and get the best possible DNA profile. Next slide. The next step is amplification. There's a couple goals of the amplification process. That the first one is to make copies of the DNA. And this is done using what's called polymerase chain reaction, or PCR. The purpose of this is to make a whole bunch of copies of the regions of the DNA that we're interested in. So since DNA is naturally double-stranded or double helix, this polymerase chain reaction process is used uses heat to separate the DNA into single strands. And then the amplification kits that we use, they include the necessary molecular items, nucleotides, primers, things like that, in order to build the DNA strand at the locations that we're interested in. And then that's going to make copies of those specific locations. And then we use a cooling process that brings those strands back together. And then now we've doubled what we originally had in one cycle of this heating cooling process. So many cycles are run, and each time those target locations are doubled. And so we're creating millions and millions of copies of the DNA locations that we're interested in. This process is done using a thermocycler, which is the picture on the far right of your screen. So the fragments of DNA that we're interested in forensic DNA analysis are called short tandem repeats, or STRs. And these are regions on the DNA strand that repeat many times. And the number of those strands, the number that those strands repeat can vary individual to individual. So since you inherit half of your DNA from your mother and half of your DNA from your father, you can have different number of repeats. And so that's kind of what we're looking for at that amplification step. And those are the regions that we're targeting to repeat, and we'll be using those downstream. So in addition to making these millions and millions of copies, at amplification, each one of those DNA fragments is also labeled with a fluorescent tag, and that's going to allow the fragment to be detected by the instrument that we're going to be using in our next step. Next slide. So this is a diagram that shows how short tandem repeats kind of work. So you can see that the blue sequence, GATA, repeats 12 times in that DNA sequence that's listed at the top. When you get a DNA report, 12 is what will be reported, because that individual has 12 repeats at that location. The bottom portion of this slide, you can see that the number of repeat units can vary between each individual. And so this is how you will start to see a DNA profile reported if you look at reports. And we'll get into that a little bit more. This slide is taken from the NIST website Starbase. And Starbase has a lot of good training materials and PowerPoints, and that's a good resource for you to go to get more information. Next slide. And this is another one from that same website that shows how PCR works. And it's used to amplify those specific STR regions with the fluorescent dyes. And so you can see the different repeats. And then that bottom is how that data is generated so we can visualize it using capillary electrophoresis, which is going to be our next step. Next slide. So during capillary electrophoresis, this is really the separation and detection phase, where what we're trying to do is now detect those different fragments that we made millions and millions of copies of at amplification. So in this step, the DNA is separated by size. And the way that that works is that DNA is naturally negatively charged. So we can use electricity to separate those fragments. They travel through a capillary that has a thick polymer inside of it. So the bigger strands actually will flow through that polymer slower than the smaller strands. And so there is a laser inside this capillary electrophoresis instrument that's going to detect those fluorescent tags. And we use a ladder. I'm going to show you a picture of that on the next slide. That's going to help us size those fragments. And then this instrument is going to generate for us what's called an electropherogram. And that's what's a representative of the data of the fragments of the fluorescent detection that the instrument was able to perform. And so that's how we interpret a DNA profile. So this is how those fragments are separated and detected based on their size. Next slide. This is an example of what a ladder would look like. This is an electropharogram, and you can see all those different peaks with different numbers underneath them. Those numbers underneath them tell you how many repeats are in that fragment. And the green bars at the top are the locations on the DNA strand that we tested for. So this ladder is representative of all of the different fragment sizes in the known population. Sometimes we have things that aren't included in this ladder, and that's part of our interpretation process. Next slide. So all of the data or electropharograms for the samples tested then gets reviewed and looked at by an analyst to determine if additional work or troubleshooting can be done to help get better results for interpretation. This is common when dealing with samples that have a limited amount of DNA present, or the quality of the sample might be low. Sometimes if that happens, then we'll only get a partial profile and won't be able to really say anything about it. But knowing how your full laboratory process works can help you with this troubleshooting phase. So you need to know how much DNA you're targeting at amplification, and to know if targeting a higher amount would help get a better profile. So that's kind of what we're doing at this step, is looking through the data and trying to use the information we have from our laboratory experience to decide if we can do more work with the sample to get a better profile. Next slide. So that's going to lead us into what are called validation studies. And really doing studies on each part of the laboratory process will help you know how those instruments and those reagents are working, and how each step of your entire system and your entire workflow is coming together to get you a DNA profile. Doing these studies are super important, so it can help you answer those questions. For accredited labs, they're also required for you to be doing these studies specific to what kind of process you're working with. So what are validations? We're going to talk a little bit about that. And a validation should be done on every part of your lab process, from extraction to quantitation, normalization, amplification, and then capillary electrophoresis. Each one of those steps includes different reagents, different lab processes that need to be looked at. And it's recommended that at each one of those steps, you include samples similar to what you're seeing in casework or what you want to do casework on. So for example, if you plan on running trace DNA samples or samples that have low amounts of DNA, specifically from groping cases or other kinds of cases, then you should include those kind of samples in your validation studies. So there's a couple different kinds of validations. There's developmental validations or internal validations. Next slide. So developmental validations are done on new DNA methods for use on forensic samples. Generally, developmental validations are done by companies that you may be purchasing instruments or reagents from. They do these validations to acquire data and determine the overall conditions and limitations of the new technique or the new instrument specific to forensic samples. So even if an instrument has been being used in an academic setting, you'll have to go through a developmental validation for forensic use, for use on evidence samples. So like I said, these are generally performed by the vendor or the company selling the instrument or the kit, and laboratories can use these developmental validation studies to help them in their internal validation studies in their own laboratory settings and environments. Next slide. So internal validations are studies that the labs do, and they're done within the forensic setting, in your specific environment, with the instruments that you have set up to do the different procedures on. So the reason for doing this is to establish methods and operating procedures for all of the different laboratory processes. And the studies generally involve looking at the performance of the instrument or the reagent. How reliable is it? How sensitive is it? Are you getting consistent results that can be reproduced? How efficient is the process? Is it going to help with contamination? What's the contamination assessment of that instrument or that reagent? How precise is it? And what are the limitations that you're dealing with specific to the kinds of samples that you'll be working on? So each lab should do their own studies on all the documentation in their lab, and this will help you with your DNA interpretation once you get your results. Knowing how all of that equipment is working in your own settings will help you when working with all different kinds of samples, but specifically trace samples. So you should, in these studies, for each different part of this study, you can create known samples with known mixtures, with a varying number of people involved in the mixture, different amounts, and then you look at that data to see what the profiles look like with things that you know what should be there. So that helps you see if you're missing information, see if you have issues, if the instrument or the process is working how you expect it to, things like that. And then these internal studies are used to define the capabilities and the limitations of the lab system and that specific process. So in addition to the developmental validations that the vendors or the companies do, you can also do a lot of these studies will be published as scientific research, and those studies, you can rely on those things to help you set up your internal validation studies. So like I said, it's recommended that you use samples that you would commonly see in casework or would expect to see in casework, which would include running samples that are complex mixtures of a lot of different people that have low amounts of DNA, start to finish through the lab, so you can see how those kind of samples are working in your system and how you can apply an interpretation, which is what we're going to talk about next. Next slide. So once you know how all of your laboratory processes are working and the kinds of samples that you're expecting to see and the kinds of results that you're expecting to see based on known samples, now you can start to come up with some studies to look at the data, which is those electropherograms. And this is going to allow you to create guidelines for how to interpret the different kinds of DNA profiles, especially mixtures, where you have multiple people contributing to a DNA sample. So those internal validation studies should also include mixture interpretation studies, and how you plan on applying appropriate statistical calculations to your interpretations. And studies specific to the mixture interpretation guidelines for the laboratory, same thing, should include samples that will be used in casework, similar to what you would see in casework. You should create known mixtures with a range of numbers of contributors, along with different amounts from the different contributors, which is what a mixture ratio is. And then these studies will allow you to look at the limitations of the different sample types, which would include trace DNA or samples that have low amounts of DNA, to see if you're getting what you expected because you know who should be there and you know how much you put into that sample. So if what you're putting in is what you're seeing, then you know that your lab system is working how you want it to. Next slide. So really the goal is to have reliable, reproducible results that are consistent from analyst to analyst. And you want to be able to know what the limitations are based on your internal validation studies. Those internal validation studies give you the scientific foundation in order to perform a DNA interpretation of the results that you get. It helps you know what you can and can't say about the different kinds of DNA profiles that you might be seeing. And as kind of a side note, the OSAC is working on a few new DNA standards that will be covering a lot of these topics. And that's going to be a good resource for direction on how to perform internal validation studies and DNA mixture interpretation studies to define what the guidelines are for your laboratory and how to go about that process dependent on what technologies that you're using. So since trace DNA samples or touch DNA samples, they're often mixtures. So having this solid mixture interpretation guidelines really is important to get those consistent, reliable conclusions that are based on that scientific foundation from your validation studies. And that will allow you to build your standard operating procedures on SOPs on how you're going to interpret mixtures based on using the data that you gathered from the validations. It's going to help you know what your thresholds are, what is a true peak, what isn't. Things like that are all part of those experiments that are designed to test the capabilities and the limitations of the system that you're working with. So knowing the limitations of your lab processes and what you're working with can help you know how much you can and should say about touch DNA samples, and will allow you to clearly communicate those limitations in your DNA reports and allow you to draw conclusions from samples like this. Next slide. So in kind of that vein, I want to talk a little bit about some of the issues that are inherent to trace DNA samples. So it goes back a few years. For a long time, forensic DNA analysis consisted of interpreting samples that had a biological fluid at serology, like blood or seminal fluid. Those samples have a lot of DNA in them, and they were generally single source, meaning there was one person there, or a mixture of two people, where it was possible to determine a major contributor and a minor contributor. But there's been a lot of advancements in DNA technologies, which is awesome. And as part of that, we've seen an increase in sensitivity and an increase in the number of locations on the DNA strand that we can test for at one time. So with the increase in the sensitivity, this has allowed us to get results from smaller and smaller amounts of DNA. And it's also increased the type of cases where a DNA analysis can be helpful, like groping cases. Next slide. But with that increased sensitivity, we also have to understand the limitations of working with those low-level samples. And while it's opened many doors to solving cases where originally there was no biological fluid present, there's also, like I said, some limitations that come with that. So one of the main not really issues, but effects of the sensitivity is that now we have more complex mixtures at interpretation. Generally, there's more contributors to touch DNA samples, and it can be caused by what's called DNA transfer. And that's something that you have to understand as you're interpreting those samples and some of the issues that exist with DNA transfer and how it relates to the relevance of the evidence that you're testing. Next slide. So I'm going to go over a few definitions. Touch DNA can refer to DNA samples that just have skin or epithelial cells, meaning that there's no biological fluid. There's no blood, there's no seminal fluid. We're working with just epithelial cells that have been left behind by somebody touching something. Trace DNA are samples with low amounts of DNA, and they encompass touch DNA samples, which is why sometimes you'll hear them touch DNA and trace DNA used interchangeably. Trace DNA is probably the more accurate term because it takes in all of it, but that's kind of the difference there. Another definition is primary transfer. This is considered direct contact with an item where the individual who touched the item leaves behind DNA. Secondary transfer is when DNA that can be passed on to an item by a person. For example, if you shake somebody's hand and then the person whose hand you shook touches a doorknob or another person, your DNA can be transferred to that item by the other person. In addition to secondary transfer, there's now a lot of studies being done on tertiary transfer, where somebody can pick up your DNA from that doorknob and pass it on to something else. So these are some of the limitations that we have to be aware of when we're working with these kind of DNA samples. Next slide. So some additional concerns to keep in mind is that background DNA might be present on an item that's completely unrelated to the crime, meaning that maybe something is found as a crime scene that somebody had touched a long time ago. It's possible to still pick up that person's DNA and they have never been at that crime scene. Contamination is more important than ever because DNA technology is getting more sensitive. It's more and more important that we wear proper PPE when collecting sex assault kits or when collecting evidence at crime scenes. Wearing those masks and gloves is super important. And then along that vein, you don't want to be talking over your evidence and restrict access to items that haven't been packaged yet. And then you're going to want to package those items as soon as you can. Collecting standards from anyone who may have touched the item is going to be important to eliminate them from your DNA mixture. And then you'll always want to be careful about what you say and understand the limitations or concerns related to the relevance of the evidence, meaning that even if someone's DNA is found on that item, it doesn't automatically make it relevant to the crime. It doesn't mean they're guilty of a crime. And there's many limitations when working with those low-level samples. It's important just to be aware of those limitations. Next slide. So now I want to go into some of the basics of interpretation and just walk through just kind of on a basic level, kind of how it works. So this is what DNA analysts do when they are reviewing that data that was generated by the lab. When working with what we call forensic unknowns or crime scene samples, we don't have any idea what should be there. So we have to do our best to figure out what information we have and what we can say. And that's where those validation studies are going to help us determine what that process looks like and what you can do with the results. So we look at the electropherograms and attempt to figure out what information we can gather from that data. So I like to compare it to a comparison that we make. It's similar to putting together a puzzle where you don't have a picture of what the puzzle's supposed to look like when it's finished. So that's why I have this picture on here. And I kind of want to use this analogy through the interpretation discussion. So in this example, it would be like having a pile of puzzle pieces and trying to figure out how many different puzzles you have in there and then put them together. So in this picture, you can see that you have puzzle pieces of 2 different sizes. So you know you probably have at least 2 puzzles in there. But then the rest of it, you have to figure out where to start. How are you going to start putting together that puzzle? And if that's the only information you had, how are you going to approach that problem? This is same thing. This is kind of where those mixture interpretation guideline studies and your validation studies are going to be helpful in finding an approach to put together this puzzle, or finding an approach to interpret this unknown DNA profile. And those studies are also going to help you know how much to say about that DNA. So DNA, like I said, DNA interpretation has become much more complicated with the advances in the DNA technology because we're oftentimes dealing with complex mixtures, have a lot of variables, and can be difficult to interpret. Next slide. So a general approach that you can take, there's an article there that references this approach that will go into more depth, but these are some good starting points for how to approach DNA interpretation. So first, what you're going to want to do is determine if the sample is single source or a mixture. You have one person in there or more than one person. And you're going to want to look at your electropherograms and look at your peaks and determine if you have any artifacts or things like that that were caused by the capillary electrophoresis instrument, any things like that that aren't true peaks. And you're going to want to determine if that's something that you're dealing with in your sample. And then you're going to want to make an assumption on how many people are in that mixture. What are the number of contributors? Because that's going to determine how you approach the interpretation. So then you're going to want to estimate the ratio of the individual contributors. Do you have one person that has a lot of DNA in there and one person that has just a small amount? That's kind of figuring out that ratio of the contributors. And then you're going to want to consider the possible combinations of the genotypes, meaning can you tell which peaks belong with which contributor? And then once you've kind of done that interpretation part, you can compare any reference samples to any portion of the mixture that you've deemed comparable. Reference samples would just be like cheek swabs, buccal standards that were collected from a known individual that you're looking for in that sample. And then as necessary, you can apply any statistical calculations to add weight to your comparison to what you're saying. Next slide. So like I said, when attempting to interpret a DNA profile, you need to be able to determine how many people are contributing to that sample and to that profile. So the more contributors that are present in the sample, the more difficult it gets to interpret the DNA profile. And then knowing how much DNA, about how much DNA you have from each contributor that's present in the sample can help you with your mixture interpretation. It can also complicate it if you have a really low level sample and there's only small amounts of DNA present. That's when we start to get into complex mixtures. Something else that you're going to want to try and figure out is if your DNA is degraded. Do you have all the information there, or are you missing some information? Do you have locations on the DNA strand where you don't have any results? Things like that are all things that are being considered during this process. Next slide. So here's an example of what we'd call a single source profile. Here you can see that there's 1 or 2 peaks at each location that was tested. Like I said, those bars at the top that are green, or in some cases yellow, those are the names of the locations that we tested for. And then if you'll remember, you get half your DNA from your mom and half from your dad, which is why some locations you might see 2 peaks with different numbers. Those different numbers, remember the number of short tandem repeats that were amplified in that sample. So that's why some places you'll see have 2 different numbers or 2 different peaks. And down there in the red dye, the red one at the very bottom, you see some that just have 1 peak. When you just have 1 peak, that means you got the same number of repeats from both your mom and your dad. So only 1 peak is displayed. So this is an example of what we would call a single source profile, which is similar to this puzzle over here that is just 1. It's not super complicated to interpret. You don't have a lot of missing information. So it's easier to look at. This is what you would commonly see from a standard, from a known individual, maybe from a blood stain, something like that, where you would have a single source profile. Next. This one is an overall example of what a mixture of 2 people might look like. Here you can see where you might have 3 or 4 peaks per location, and you can see that there's a difference in peak height. Some of the peaks are tall and some of them are smaller. And so with proper interpretation guidelines, you can start to figure out which peaks go with which person. So maybe all of those tall peaks might be your major profile, and the smaller peaks might be your minor profile. Next slide. So this is a close-up of that same profile just to help you see it a little bit better. Also with a puzzle next to you, see that fire truck puzzle has some other little pieces on there. It's easy to see that you have 2 puzzles that you're working with. But the bottom one, in the bottom right corner, there, all of those puzzle pieces look to be the same size and the same color. That's going to be harder to tell who's who. So like I was saying, where you can maybe say all of the tall peaks belong to the same person and all the small peaks belong to another person. Sometimes you're going to have 2-person mixtures where all of the peaks are about the same size. And then you can't really tell who goes with what. And that's starting to get into more of a complex mixture situation, what we would call an indistinguishable mixture, where we can't determine who is who. Next. This is an example of a 3-person mixture. You can start to see the more people you get involved in these mixtures, the more peaks you have to work through, the harder it can be to determine if you have true peaks. So on a lot of these, you're going to see 5 or 6 peaks per location. That's what's going to indicate that you have at least 3 people in that mixture. Next slide. This is a close-up of that same profile. Hopefully that allows you to see it a little bit better. And same thing with all of the different puzzle pieces. Sometimes it's easy to tell what your small pieces are, or your minor profile. Sometimes it's not. And sometimes you can, depending on your interpretation guidelines, you might be able to pull out a major profile, but then when it comes to the smaller peaks, you can't tell who goes with who. Who's the second contributor? Who's the third contributor? How do those peaks match up? And this is where all of your validation studies and all of your mixture interpretation guidelines and your standard operating procedures really come into play to help you decide what you can and can't say once you start to get a lot of people in these mixtures. Next slide. This is an example of a 4-person mixture. Same thing. You're starting to see a lot of peaks here. And you're going to start to see some that are really low, some that might be some places where you might be missing information. So 4-person mixtures, it gets a little bit harder. Some labs will interpret 4-person mixtures, especially if they've validated what's called a mixture deconvolution software that can help figure out who could be who, and it will give you different statistics on the different genotype combinations and possibilities. So it's really going to come down to the lab onto whether or not they'll interpret these mixtures that start to have a lot of people in them. So that's another reason, like, we encourage you to work with your local laboratories, figure out what they do, what their interpretation guidelines are, what their capabilities and limitations are. Next slide. This one is an example of a partial profile. You can see, looking at this, you have some peaks that have a number underneath them and some that don't. The ones that don't would be considered below our analytical threshold. That means that below a certain threshold, it's not something that we can confidently call a true peak. And same thing, that goes back to your validation studies and what you've seen, what you do in your lab. But these are partial profiles where you can see that you have a lot of missing information. So it's like getting a puzzle with only a few pieces. So you know that you don't have enough information there to put together the full picture. And it can be hard to determine how many people you have contributing to a mixture like this. For us, like, this would be considered currently like an uninterpretable mixture. We're missing a lot of information. Maybe we don't want to say a lot about it because we don't know really what's going on. There's not enough DNA there for us to confidently say something. Next slide. So that brings us to some statistics. Once you've gone through your mixture interpretation guidelines, you've decided what kind of mixtures you can interpret, what you can and can't say about them. If you make an inclusion or you make a match to somebody, you can add a statistic to that to add weight to that conclusion. When it comes to working with low-level DNA samples, trace DNA, touch DNA, like I said, we're oftentimes dealing with complex mixtures, and there's different softwares, probabilistic genotyping softwares out there that can make working with those complex mixtures a little bit easier, a little bit better. It just really depends on your validations and what your lab does as far as their statistics. Something to keep in mind is that matching the DNA to a specific individual doesn't automatically make it relevant. Like I said, there's many limitations when working with trace DNA and those complex mixtures. You can have background DNA, transfer events, things like that that you have to keep in mind when dealing with this kind of evidence. So there is great value in being able to work with smaller amounts of DNA because it is very helpful in specific cases like groping cases, where now we can get information where before we couldn't. And that's really helpful in these kind of investigations and these kind of cases. But it's also important to understand the pros and the cons to working with trace DNA and complex mixtures, and then also explaining that to a courtroom and a jury. So it's necessary not just for DNA analysts, but also investigators, attorneys, judges, potential jurors to understand what the capabilities and the limitations of the DNA evidence are. And then from a forensic laboratory perspective, it's important to have those validation studies and scientific foundation to help you work with these kind of mixtures to be able to add value to the cases that we're working on, but to also understand the limitations. Next. So to kind of wrap things up, we want to bring it back to the original case study review that Julie talked about in her original webinar, and revisit that groping case study. The groping sex assault case, specifically the victim was attacked outside near her vehicle in a parking lot, and the perpetrator grabbed her in several locations on her body and her clothing. And in my opinion, the case was successful for a few reasons. First of all, the victim deserves a lot of credit. She was able to give very specific locations where she was grabbed and where she was touched. So that helped guide the forensic nurse to use that information, to collect that evidence and think outside the box as to where would be important to collect specific to this case. And she was able to collect and document that possible evidence from the perpetrator. And then the collaboration between the forensic scientist, the forensic nurse, and the police detective were key to collecting additional DNA samples from the clothing and processing those samples through the lab. And by having those exact details, we were able to collect from specific locations, limiting any potential background DNA, things like that. And we still had mixtures, but by being able to target those specific locations at collection, we reduced the likelihood of getting mixtures that were too complex to interpret. And this helped create the touch DNA form that Julie mentioned, that now allows for that communication to be easier and more consistent with the documentation between the nurse, the scientist, and the police detective, with the hope that the better collection from the trace DNA samples will help with the interpretation and the lab work in these groping sexual assault cases. Next slide. That leads us into the importance of collaboration, working together. Everybody has a different focus, a different specific task, but when we come together and we're able to help gather information and see things from a different perspective, it helps make the investigatory process better, ourselves better, and our organizations better, and it helps us be able to learn and work together with that wide variety of disciplines, which not only makes our job rewarding, but helps and can help solve some of these groping sex assault cases, as well as other cases that involve trace amounts of DNA or touch DNA. Next. So we want to thank you for participating in the two-part webinar series with us. We appreciate your participation. If you have any questions for us, we can go to the next slide and ready for questions.
All right, thank you so much, Heather and Dr. Valentine. We do have some questions that have come in. So the very first one came in early in the presentation, asking about swabbing clothing, such as when a patient does not want to surrender the garment.
Yes, this is Julie, and I can address this. On the case study, in that case, swabs were collected. So this was the first time we had never collected touch DNA. Only possible way of getting any DNA of this assailant. And so the forensic nurse used 2 moistened swabs and actually swabbed the clothing areas where the victim stated she had been touched. And it actually was a pair of those swabs that developed a full STR DNA profile. But she also allowed her clothing to be collected. We believe the best practice is to send in the clothing, but if you have a groping case, and we really have presented this to our transportation police, because sometimes they have more groping cases, airport police, etc. If there's a groping case where the victim says, I don't want to give my clothing away, then in that case, I would use 2 moistened swabs. And just as you do for skin, you would swab that clothing and note where on the clothing you had swabbed and that the victim had declined to give the clothing. With that, I will also say, when we have groping sexual assault cases, we still respond with a advocate. We have Rape Recovery Center or other centers that we use that we will have an advocate because the trauma experienced by the victims in groping cases, some might think, well, it's not a completed assault, but it can be just as traumatic. And so we will have advocates that will bring clothing for the victim so that they can have replacement clothing if they do want to give that clothing.
Okay, we do have another question that came in asking, with such great advances in technology and sensitivity, will this not make the detection of pertinent DNA more difficult and also maybe create problems in court?
Yeah, this is Heather. With that specific question, the answer is yes, it can. But like we mentioned, you also need to know the limitations and be able to explain the relevance of your evidence and explain to the jurors your process. So I think we touched on that a little bit there at the end. Like I said, it can, but by knowing your validations, your studies, your interpretation guidelines, having that scientific foundation, and knowing how to explain those limitations in court, I think is really important when working with these kind of evidence or pieces of evidence.
All right, next question we came in right after that, so probably along the same question line from Kimberly asking, in your specific lab, when analyzing minute samples from sexual assault groping cases, which technique yielded better results, robotic or manual?
We've actually validated both for all of it. Our pathway is a little bit different right now in that all of our sexual assault cases with the potential for seminal fluid go the robotic route. And then groping touch cases go the manual route for now. But we have validated the instruments to work with all of the different kinds of samples in our lab, regardless of if they have a fluid or not, if it's a trace DNA sample or not, it can work with either pathway. We just split it up currently based on our laboratory flow and kind of our case needs and the way we have our different teams set up.
All right, we are going to give everyone just a couple of more minutes to type in any questions they might have. You do have a great comment from Robert, who said that this was an excellent follow-up to the first webinar and said thank you, asking what is also in the future for Touch DNA.
From a laboratory perspective, I think the future for Touch DNA is pretty exciting when it comes to new technologies that are coming out that are more sensitive, that can give additional information than the locations that we test for now. There are new technologies that are coming out that will allow us to look at even more locations, places, SNPs, not just STRs, different things like that. And I think that there's a lot of studies that are being done as far as touch DNA, the effects of transfer, things like that are coming out pretty often that are exciting to kind of see how things are looking in casework. What are we able to say with these kind of samples? And a lot of those studies are being done both here and other places to look into those results and the effects of transfer on touch DNA samples and the evidence, the relevance of it.
All right.
And I would just add on there from a multidisciplinary perspective, I think a big focus right now, I have heard from some jurisdictional areas where there might be some hesitation to go this route and start to collect DNA, touch DNA in sexual assault and groping cases. I believe that our case
All right, we're just going to give everyone a minute to see if any other questions come in while we are waiting on kind of the last questions. Before we end today's webinar, we do want to make everyone aware that we have recorded today's event. We will be converting it into our archival version over the next few days. Should you want to go and refer back to any of today's presented material, or if you have any colleagues that might have missed today's live event, you can pass that along to them, and they can then watch the archival recording. We do also have back on the main page all of today's reference material that was provided by our presenters. We have a presentation slide copy for today's presentation, as well as the first webinar that occurred back in February of 2018. So you can download both of those PDF versions of the slide decks before you leave today. Dr. Valentine also provided a few other reference materials that you can find there in the back on the main page. Dr. Valentine, do you want to touch on those reference materials real quick before we do close out today?
Certainly. One of the reference materials is a list of literature that might be helpful regarding touch DNA. The other reference is our State of Utah Touch DNA form that we've done education with a multitude of law enforcement agencies across the state, and is part of our electronic forensic electronic medical record that we use to help guide our evidence collection, as well as inform the forensic scientists on the areas of collection to help them in their analysis decisions.
All right, thank you so much. And with that, we are going to go ahead and close on today's event. First, we would obviously like to thank both of our presenters, Dr. Julie Valentine and Heather Mills, for their presentation and all of the work that they have done in the touch DNA field. So thank you both for joining us today. We would also like to thank all of our attendees for joining us for today's webinar. These webinars are just simply not possible without everyone's attendance and support. So thank you all for joining us for today's follow-up webinar in the Groping DNA subject matter. Before you leave today, we always ask that you participate in our short survey. You'll find that directly below on the right-hand side below the closed captions. Simply click on the word survey, and that will open up a new window for you where you can give your feedback about today's webinar as far as the webinar experience, as well as the way that we disseminated that content out to you, our audience. So taking a few moments to complete that is valuable feedback that we always look forward to receiving from you. You will also see in our chat pod, Tyler will be posting some links to our social media outlets such as Facebook, LinkedIn, and Twitter, as well as links to our podcast and our website where you can find all of our no-cost webinars, events, success stories, and reports that we are constantly updating and putting out to you, the forensic community. Again, thank you so much to our presenters for joining us today and your presentation. Thank you, everyone, for attending today's webinar, and we look forward to seeing you in our next event. We hope you have a great rest of your afternoon.